Hello everyone, welcome to BrainMap. This seminar series is co-sponsored by the Peabody Funded Center for Mesoscale Mapping, housed in the Martinos Center. Sorry for the little delay in the beginning of the talk, and still we managed to figure out. And so we are great to have Dr. Firas Mawase today. Dr. Firas Mawase is an associate professor of biomedical engineering at Technion, Israel Institute of Technology. His lab investigates the mechanisms that underlie motor behavior and the recovery of the movement after brain injury. He earned his Ph.D. in biomedical engineering from Ben-Gurion University of Negev and completed postdoctoral training at the Johns Hopkins University School of Medicine. He's currently on sabbatical at Harvard University and MIT. I'd like to remind our audience to address your question at the end of the talk. Without further ado, stage is yours. Right.
Right. Thank you so much for hosting me, and it's my pleasure to be here. So today we'll be talking about a few projects that we have been doing in the last few years, and basically all around the topic about how does our brain control the finger movement and hand dexterity. So I will be using lots of studies of a human patient after stroke, and computation modeling, and also in the last part of my talk, I will be discussing some new findings about non-human primates where monkeys trained to do some finger movement, and we record from different areas in the brain and try to understand more about how our motor system sends commands to control finger movements. So the big question that we are asking in our lab is how is neural activity organized to generate dexterous finger movement? And as I said, like, we use different approaches. Today I will be focusing in the first and the second and the last approach, which is basically focusing on how lesion due to stroke affecting the finger movement. How we can map brain activity in healthy participants while participants can perform some of these dexterous movement inside the scanner, and what's the correlate between this functional mapping and then some measures of hand function. And lastly, I will also, as I said, I will talk a little bit about more monkey stuff, doing some trained dexterous hand and finger movements. So I think why I really care about dexterity from two reasons. First is that we know that from the clinical perspective, lots of stroke patients actually their hand function and fingers are really impaired after stroke. And they recover for some extent, but they never go back to the same level at their baselines. So finger dexterity is really one of the long-term disability that these stroke survivors face when they're living their normal living or their life after the stroke event. So the loss of this dexterity is actually limits our independence, our living the normal life that we used to live after that big event happened into our brains. From the second reason is that more about the engineering aspect of it, which is there is currently a really large gap between our real hand functions and the most advanced BCI that somebody ever developed to try to decode some of the neural activity and to send that command to the bionic hands and perform finger movement. All right, so if you just compare very simply about how many degrees of freedom we have as a human in our hands and how much is the best current BCI has. So 25 degrees of freedom that we can do in our fingers, different joints, different directions, different forces, and the most advanced is 4 degrees of freedom. This is one of the last actually studies published in Nature Medicine a few years ago in a group from Stanford where they decoded some neural activity from the human primary motor cortex to try to control a BCI using this neural activity. And if you also look at the speed of that movement of the real hand versus the BCI, we can see that there's a big gap between this kind of like how fast we can perform our finger movement, which is fast as 200 milliseconds, versus the best movement that this BCI can perform is up to 1.6 seconds. All right, so there is really a big gap between this kind of hand use and the BCI use. And I think the gap is actually because we still don't fully understand how the central nervous system control our finger movements. All right, this is the gap as actually can be summarized by that. And so today we'll be talking a lot about how we measure dexterity and what I mean when I say dexterity and hand dexterity. All right, how stroke affect that dexterous movement and hand function, how we can link between lesion location size and the hand dexterity, doing some kind of like lesion mapping of the lesion and the behavior metrics that we measure in our devices. And then try to more like this is more about the macro scale level and about some network level between structure areas and the net and the behavior. And then I will show like how artificial networks that are trying to mimic some of the behavior deficit that we observe, for example, in stroke, looking at the neural activity of that artificial neurons. Can we get some insight from that kind of like, dynamics of these cortical neurons? And then we can test them further by recording from biological neurons from non-human primates performing very similar finger movements, trying to make that link between activity and behavior. So this will be basically the basis of my talk today. And when I say dexterity, right, what I mean by dexterity, I think this is like these are many examples of daily life functions that show what I mean by dexterity. All right, so all of these examples from tying shoelaces, playing any musical instruments, even dressing, eating, typing, tapping, all of these very fine control hand functions is actually a dexterous movement. If you really look closely at this movement, so it's not only about producing force of end effector, which is our fingers, but also it's more beyond that. It's more about digits or muscles that need to stabilize your hand when you perform some of these functions. There are also other digits or other muscles that need to be suppressed to prevent some unwanted movement when you perform that kind of like finger dexterity. All right, so it's a combine. It's more about precise coordination of finger movements that allow these kind of like dexterous hand movements. All right, so that's just producing force. It's coordination between different fingers to allow this kind of very precise and sophisticated finger movement. Now, this ability is really unique to human, right? If you just look at the evolution of that hand dexterity and the ability to perform that dexterous movement is very different across species. If you compare rodents, higher mammals, monkeys, for example, and a human here. All right, so rodent has no ability to make an individuated finger movement, for example. All right, monkey still has some ability to make, for example, pinch precision, to like have this kind of like opposing thumb and index finger to hold some of these force pellets and then food pellets. I'm sorry, but none of these pieces actually can perform this kind of very simple pointing movement. But even like this simple movement is very unique to human. So we know that humans really has this kind of like, I would say, advanced and sophisticated ability to make that precise coordination of different muscles to allow us making this dexterous finger movements. So how we measure dexterity in humans? This is one of the biggest questions that when I open my lab, I started to say, well, how we measure that, right? We need some kind of like a simple device that can measure dexterity. And the idea is that we develop a hand device or dexterity device where we can really measure in high resolution forces in different directions for all digits simultaneously. All right. So the idea is very simple. We have two first sensors here. One of the sensors can measure the forces in the flexor direction and the flexion direction, and one sensor that can measure force in the extensor direction. So all what I talk about is isometric tasks. So there's no really kinematics moving. It's more about reducing forces in different digits and different directions. All right. This is like it's a better way of quantification, this kind of like dexterity. And participants can really, it's like a video game. They can watch the screen. They can see that in this try, they're instructed to move one digit on a time. For example, here they requested to move the index finger, and they can actually produce different force level based on their maximum forces. So 20, 40, 60, or 80%, and then they push down the force sensor, and then they can control the cursor position going up and down in the extension direction or down here in the flexion direction. All right, so very simple task that we can ask participants to perform. And this is how the force profiles looks like in the flexion and the extension direction. This is here, this kind of like purple magenta color is the instructed digits, and this is the force profile of all the other digits in one hand. All right, so we can easily perform that task, and we can follow the instruction and hit the target of that force level. So how we quantify dexterity from this? What I would like you to emphasize here, I'm giving you two examples of two different trials. I think this is important to understand how we define dexterity or ability to make individuated finger movement. This is the critical part of this behavior measure. If, for example, like if you see this kind of like a single finger task where the subjects were instructed to move the middle finger, all right, and to make sure that all the other fingers are immobile, or they are not moving. All right, this is not an easy task because you are controlling all of the force of your 5 digits. And if you look at the low forces, we are really good at hitting the target here in the red rectangle, red target here by producing force only in 1 digit. There's a little of other forces in the other digits, but they're really small force levels, right? But now when we start to ask participants to produce more forces in the task, so it's the same task, but just you need to push harder on that key, in that sensor. And first you see that, well, we are good at hitting the target, right? This is the instructed digit, and this is the target location, or the target, the force target, and they can reach that force target very easily, right? But then the point is that here, if you look at the forces of the other non-instructed digits, you will see that there's a lot of what we call enslaving. So there's a lot of forces actually preventing, making that finger individuation our pure finger individuation. All right, all of these non-instructed forces, forces of the non-instructed fingers. So the way that we quantify that, well, we said let's look at the relationship between this kind of unwanted forces or enslaving in Newton as a function of the force that we produce in our instructed finger. All right, and this is exactly what I'm showing you here, is the enslaving of the unwanted fingers and unwanted fingers or digits to instructed digits as a function of the peak force of the instructed finger. All right, so there's two things you can observe from here. There is a really large differences between extension movement and flexion movement in blue here, right? And one more thing is that if you look at the slope of that actually relationship, this is really a measure that gives us somehow a metric of how we are good at individuating that specific digit. All right. If you have really a zero slope, there is no really slope of this line. This means actually that there is no enslaving whatever force we apply in that instructed digits. All right. The more the slower the slope is higher, the more enslaving we have given a specific force that we produce in that specific task. All right. So the slope of the slope of these lines actually is really a good measure of the individuation ability, and we call that individuation index. And you can do that for all of the digits in both directions, and you can come up with some conclusion about what exactly is the differences in the behavior level between flexion and extension, right? So one of the things that we observed here significantly across all digits is that, as I said, we are much better at flexion than extension, our fingers. And there is also digit differences, something that we expected actually. Our thumb and index are the mostly individuated fingers and controlled compared to the other third, fourth, and the fifth fingers. The worst finger in our hand, activities is actually number 4, the ring finger. This is the less individuated fingers. Wherever we ask to move that finger, we usually apply much forces in all the other fingers. All right. So we quantify this dexterity as a measure, of the ability of people to make that dexterous movement. But then one of the concerns we actually face is that if you look at the finger individuation, so it's actually an outcome of at least two different factors and independent factors. One of them is about, the neuromuscular commands that are sending that commands to the muscles to perform that specific task, right? But there are also differences between how the biomechanics of the hands looks like between flexion and extension. There's a lot of muscles, tendons, soft tissues that actually are different between moving to this direction or moving to that direction. And we ask a very simple question. So might be the differences that we observe between flexion and extension movement is actually driven by some mechanical coupling of peripheral factors, nothing to do with the differences at the neural level, controlling that different aspects of our dexterous movement. Right? So to control for that, my students UL, they develop a passive task where now, instead of like asking participants to actively, voluntarily move that fingers, now the robots move all of these fingers. And we measure how much enslaving we have for that passive movement due to the mechanical couplings. And we made sure that there is no really any active movement here because we measured also EMG activity, making sure that participants were at rest, and what actually driving this movement is basically the robot that I can show you here, a short video about that, how this actually movements looks like in the robot. So now the robot has 5 different motors that can move digits in different directions. So here, for example, the index finger in the flexion, and then the ring finger in the flexion. At the same time, we measured all the enslaving, all the forces of the other digits. And what UL found is that actually very striking for us is that enslaving due to mechanical coupling, like the differences between mechanical couplings actually was much higher in flexion direction. So we have more actually biomechanical couplings that actually preventing us to make individuated finger movement in the direction that we show that they have much better dexterity when subjects voluntarily make that movement. All right. So if we go back to the same simplified model of that kind of like, finger individuation is a sum of some neural input and mechanical coupling here. And the fact that individuation is much larger in flexion and extension, and the mechanical coupling is larger in flexion versus extension, this suggests for us is that should be some differences between the neural control level of finger flexion versus finger extension. Yeah.
This is handedness you have there, right? Is that?
No, here is it's the hand. It's a hand level. So we average across all digits just to, how, because we see the difference is very consistent between flexion and extension across all digits, and we just summarize that in a hand level.
What about handedness? Because I just realized on my right hand I have more. And I am right-handed. I can, for example, fold my pinky kind of independently, but I can't do it on my left.
Right.
So is that something that?
It's a good question. So all of our participants are right-handed, and we tested them in the right hand. So and we have a lot of pilot data and other studies that showing that there are some differences, but they are very subtle. There is no large difference between right and hand for dominant and non-dominant hand, but dominant has a little bit much larger dexterity ability than non-dominant hands.
Musicians, yeah.
Well, yeah, we tested that. Musician has really larger dexterity compared to healthy controls. Naive non-musicians people, right? Absolutely.
Mechanically also.
The
mechanical.
Mechanically, a little bit less enslaved, but the difference is what we observed. We think it's more about the neural control because the difference in the individuation index cannot be explained by the small changes between the mechanical things. It's more about the neural control of these things. Yeah, but this is we have that. We didn't publish this data yet, but yes, it's a good question. So as I said, like there is something interesting difference between how we control or how the motor system controls this kind of like dexterous movement between flexion and extension. So now let us, we were thinking about, well, how to study that, how we can really correlate between some activity or some structure or functions between areas in the brain and finger dexterity. So one approach was using stroke patients. So far, we recruited 45 right dominant ischemic subacute stroke There are 40 days after the events. We also have age match and gender match controls. And what I'm showing here is the overlay lesion mapping of these stroke populations. And as you might see, there is a lot of impairment or lesion at the cortical spinal, M1, the internal capsule, the precentral, and also the corona radiata. 45 participants is not that much participants. So at this point, for me, like it was really hard to make any, compare. We don't really have enough power to compare between right and left strokes. So from now and so on, we flip the lesions and we making, some correlation between lesion locations using some techniques that I will show just in a bit, and deficit in the finger movement. And the way that we measure finger individuations using the same metric, the same device. We use a robot here in this experiment, where stroke participants were requested or asked to move single digits on a try. Right? So we measure that forces. We measure the enslaving. Of course, there is differences between the strength between stroke and age control. We control for that differences in the strength, and we measure these finger individuations or the ability to make dexterous movement between stroke in the bars here, and this is the control population in the dashed line between flexion, glue, and extension in red. All right. So there is one observation here is that both directions are below the controls, right, in all digits. This is something interesting that, we see that stroke patients are affected in their flexion movement and also in their extension movement. So there is a really significant between controls and stroke patients in both directions. But then when we actually normalize these kind of like deficits based on the control ability to make finger individuations, and we call that metric as impairment, which is basically how much actually the stroke patients in the extension direction versus the flexion directions were really impaired compared to the healthy controls. All right, and what we found that, well, of course, both are impaired, both they are not larger than 0, right? But if you compare between within subject comparison, you see that these patients are much affected in their extension movement, like to open their fingers versus to close their fingers, for example, to control in the finger individuations in the individual finger level in the flexion direction. All right. So then so we took this behavior measure and we try now to correlate between the lesion location and the deficit at the behavior. And this is a very simplified explanation of how we did that. So we have a lesion for each patient. We detect the lesion location. We mark that, and we have N participants, 45 in our case. And we go, we went voxel by voxel and quantify how many participants were affected in that specific voxel. All right. So we can get up with a lesion load map of, darker red here means more and more participants were affected on that specific voxel. Right. This is kind of how we define the lesion load, and then we correlate that lesion load with the behavior, with the finger individuation dexterity index that we have, and then we can get a statistical map of correlates between deficit at a specific condition, specific movement, and the lesion that we have in our stroke patient. All right. So, and then we can look at, well, where is that lesion located? And I was really curious about, there's a lot of different areas that were affected. But for me, like looking at some I would say correlates with the finger movement or any motor functions. So really curious about a very predefined tractographies that we know that they are involved somehow in the producing some of these finger movements. Okay, one of them is the M1 cortical spinal tract, that actually descending pathways from the cortex, the primary motor cortex, down to the spinal cord. The other one is the premotor area. It's again descending pathways that start from the premotor area, Brodmann area 6, and also goes down through the cortical spinal tract down to the different subcortical and down to the spinal cord. And then you can ask question, what is the correlate? Is there any significant relationship between this lesion and the deficit that we see in the behavior, and whether that deficit was more biased toward only flexion movement, extension movement, or actually deficit on a specific voxel affected both sides in the same way. Or this is basically the question that we asked. This is axial slices of these of the MRI scans, and here what I want to show or emphasize is that when we look at M1 cortical spinal tract here in cyan color, we found a few things. We found that both flexion and extension actually are affected. So there's a bidirectional effect in the dexterity when there is a lesion in these kind of like areas. All right, you might see some biases toward few voxels that survives all of these statistical comparisons that are located in M1. This red here is the extension, blue is flexion, and this purple is actually a bidirectional effect on the behavior tapes. All right, so within M1 cortical spinal tract, we observe that both flexion and extension are affected, a little bit biased toward extensor movement that were located in the M1 hand and nope. Looking at this kind of like lesion map. But then we start to look at different tractographies. For example, the premotor areas, we do observe some biases toward more flexion hand dexterity, flexion individuation index in the flexion directions. All right. If you see like this is a green premotor cortical spinal tract, and that actually was more overlap with lesion that were voxel of lesion that were associated only with the flexion movement. All right. And then we also can go further and look at different tractographies that we know from animal studies that any lesion in these tracts might affect finger movement. But here, like it's kind of like a nice sanity check that these tracts are important. There is a lesion load with these tracts, but there is also some biases. If you look at the really small details where that voxel associated with flexion movement is located versus voxels that associated with extension movement are located. Right? To summarize this in a really kind of like a schematic image is if it's like a slice of M1 area 4, area 6, and the somatosensory area 3, you would appreciate that for motor neurons, for example, driving some muscle activity in the extension direction or driving muscle activity in the flexion direction to produce that individuation index, we see there's really a lot of like inputs, similar inputs coming from M1, S1, CST toward both directions. The larger biases that we observe for flexion movements actually was driven by more premotor areas driving this kind of like motor neurons. So this is, you see, like there is a really gradual shift towards blue lines here versus a more mixed population that we observed in the M1 cortical spinal tract that drive these kind of like differences in the finger individuations. So this is one really exciting result that for years the idea was that there is no really biases in the human primary motor cortex between flexion and extension. People did functional MRI for a very simple single finger movement, and they observed very similar representation of that different directions. So it seems that and we replicate that, but it seems like representation of flexion movements is a little bit larger, and it actually goes to beyond the area 4 and the primary motor cortex, and actually it touches also some premotor areas that can somehow control some aspects of finger individuations in the flexion direction. So I mentioned finger individuations is really a good measure of dexterity. Larger individuation index means that you are really controlling that specific digits much better than other movements, right? But there is also some weakness on that. There is some limitation in this measure, and I will just emphasize this here. This is how one trial is an example of one trial for healthy participants where he instructed to move the ring finger in this trial. All right, you see very nicely he flexes his ring without that much enslaving in the non-instructed digits. And you know, if you look at the individuation index of these two actually patients, you might see that they're really close to each other. They are less than their controls, but they are 0.75 versus 0.8. So if you really just look at this kind of like average individuation index, you might think that these two patients actually are impaired similarly, right? But then if you look at the force pattern of these two patients, there's a really differences between how they produce forces in the different digits. And our initial individuation index cannot really quantify or account for these kind of like differences in the force patterns across all digits when you perform this kind of a task. All right. So how you really can quantify this kind of like similarity or dissimilarity of the force profiles given a specific trial in a given in a specific force that they were requested to produce. So to do so, we went to functional PCA analysis, where the idea is very simple. The idea is like to define like a low dimensional PCA's space for our healthy controls, and then to project your stroke data on that space. All right, you don't really need to measure any endpoint errors or smoothness of movement. You just project all of these force profiles. So there's no really prior assumptions about how your data should look like. Right, you just define the reference healthy control population, and then you project all of these stroke patients, stroke data there, and then you can measure the distance between your clinical data and the reference populations. And the idea is that smaller distance means that my force patterns are really quite similar to how would healthy controls produce that specific condition. All right, larger distance, we use Mahalanobis and Technion because we want to also take some account for the covariance between these two metrics, and there is covariance. So Mahalanobis is a better index that really give us quantification about how similar or dissimilar the force patterns of the stroke versus the healthy controls. All right, larger distance means very dissimilar. Right? Zero distance means that you're really on spot of the mean of the reference population. All right, so and this is very interesting because actually it gives us some insight about the clinical deficits of finger dexterity when we really account for force level and direction during this dexterous movement, something that we couldn't really draw when we look at the average individuation index. And what we observed here is like when you look at the Mahalanobis distance between flexion and extension, blue and red, as a function of force, as I mentioned before, you start to see that, well, there is interesting and significant interaction between this Mahalanobis distance or this similarity and the amount of force that patients were asked to produce in that specific trials. Right? And what we observe here is that at low forces, there's a really larger deficits in our stroke patients in a way that their force patterns are really largely dissimilar than flexion and than healthy controls. All right. If you go and for me, like yes, there is deficit in all of our movements after stroke, but this actual deficit is dependent on the amount of force that we apply. All right. So if you look at for me, like a more conclusion from this kind of like results is that we have a general deficits of dexterity, and these deficits actually is uniform across force level. But then on top of that, there is some direction dependent that we observed here in the extension movement if you go to low forces. This is really important also from the rehab perspective, that if you really want to think about, okay, how we want to improve dexterity in patients and stroke, and whether do we really train our participants to do more extension movement versus flexion. This is one approach. And this results actually suggest that, well, yes, we need to focus more in the extension, but you also need to focus much more and spend more time and effort in low forces where these patients were really impaired compared to their other flexion and the healthy controls producing the same task. All right, so this kind of like direction-dependent things were really exciting for us. That what we know our measure can give us a little bit clinical insight about how these differences between flexion and extension and what the relationship with the force. So to directly test this hypothesis, we went back to healthy controls and we used some kind of like interference of the neural activity using a transcranial magnetic stimulation online while participant performing. We said, let's start with a simple task, just the index finger, flexion versus releasing the force for low forces versus high force level. So similar task, we measure the strength, and then there's a tracking of the force target in both direction, force generation in the flexion, and then releasing. And then we apply TMS in 3 different areas, in the contralateral M1, the ipsilateral M1, and as a control in the PZ direction, just to test, is there really any relationship between interfering with that specific element of the performance during this kind of like finger flexion releasing at different force level, right? So participants, really, it's a very simple task. They need to push down and release that force, but it's not like it's a controlled release. So they, like during the release of their forces, they need to track that target on the screen. And in some trials, they have a larger force. In some trials, they have a lower force, 20% versus 60% of their maximal forces to contraction that they can apply. And we measure behavior in terms of like accuracy and the smoothness of the movement as a measure of how they are good at this kind of like task. So the first results was that interfering with during performing the tasks when that TMS was applied in the contralateral M1 has a really a larger effect on the performance versus ipsilateral and without TMS. Everything was normalized here to the control site, which is the PC. All right, just to control for the sensory feedback of the loud sound of the TMS, and also the sense, the touching sense, and also for these kind of high frequencies for 2 seconds to control for that, we applied in the control sites. But then when you look at this kind of like how these changes are actually depend on the force level and the direction, we did find a very interesting interaction between direction and force levels. So TMS interfere with this task was larger when we applied low forces, and when we release our force, then producing force in the flexion direction. So it goes actually back to the kind of like interaction we found with the stroke, right? So we found that in lower forces at extension, we have much more deficits in the finger individuation or the impairment that we observe with this Mahalanobis descent. And it seems that when you interfere, like causally interfere with that without kind of like skills during practice, you see that there is larger effect at low forces and at the extension direction versus the other components, right? Both components are affected, right? But just there's interaction, so larger effect on the extension versus the flexion movement. And so from the rehab now perspective is that can we change that dexterity? Can we improve it for somehow, right? And this is really was a kind of like a challenge for us, because we do think that dexterity and the way that we measure kind of like this dexterity is kind of a quantification of at the impairment level. So these patients are impaired to perform that finger dexterity movement, and individuation index and all of these Mahalanobis multi-dimension measures give us really precise quantifications of distal muscles and distal movement of the hand. And this is a study that we did back when I was a postdoc at Hopkins. We asked, can we really make a rehab program using intensive training of kind of like chord tasks, so kind of like piano, using the same device, but now participants were actually trained to do this 2 and 3 fingers chord task and whether that improved their dexterity. All right, so it's intensity. There's a lot of like kind of like a visual rewarding system working here, because if they are successful, they get reward with a score and pleasant sounds. And to make that long story short here is that this even though at the chronic stage, so defined as a 6 months after the event, we could improve their dexterity level by kind of like this intensive piano-like core task. All right, this is the individuation index, and that improvement actually generalized to more functional tests. The ERAT is like one of the clinical assessment of hand use and hand functions in stroke populations. And we did not train on that, of course. We just measured pre and post training and post 1 week, 1 day and post 1 week. And we found that this kind of like intensive training can be improving their impairment and also long lasting, at least for 1 week after they finish this kind of like study. All right. And then we asked, well, this is a chronic phase. If we can start early, for example, subacute phase, like I would say a month or something after the stroke, can really improve that dexterity. And it's still an ongoing study. But so far, we have 22 participants, subacute. They trained for 10 days, like 2 weeks, roughly. And we found that, we targeted the extension in the low forces here. So you can see that actually this kind of like training, it is an intensive training, I would say, like it's a 150 tries a day. We can reduce that kind of like abnormality of the extension movement at low forces that we observed before in our stroke populations. So it's something that we can target, we can reduce it. But just to be clear, we never succeed yet to go all the way back to the 0 levels. So it's really hard to make a full recovery of hand functions, but we do improve their impairment a little bit. And that improvement generalized to these patients' everyday tasks when they eat, dress, and do all of these basic functions. Right? They're still limited, of course, but they are much better than before the training that we induced in this 2 weeks of program. All right, I have 2 more studies. So it's nice. It's like implications about some of the, I would say measures, some of our insights about how the brain controls extension, flexion versus low and high forces. We kind of like, targeted that question from different methods and different experiments. And for me, like really puzzling for me was what happened, as I said, at the neural level. What happened at the single cells activity level? Right? If you look at the motor cortex, well, of course, we are not there yet. I have no idea at human what they are doing, what is the single cell or the population level are doing to allow this kind of like dexterous movement. The lesion mapping things can link between areas, like a bigger structure, macro scale level, and the deficits, but they're not really good at telling what happened at the single cells level or even at population level happening or doing something during inside the motor cortex. So one way to kind of like close that gap is to look at, or if we are able to develop some artificial neural network that with the prerequisite is that this network can really mimic the behavior that we see in human. If that network is good at replicating most of the behavior stuff that we observe, then we can go and look at what these artificial neurons are doing to understand, well, that kind of like very interesting dynamics happens, allowing this kind of like specific finger movement, for example. Right? You can infer some understanding about how this neural activity happening. And what we did here in this study with Ashraf is he started with a very simple artificial neural network that he built it with 3 different layers, only to control 2 fingers, 1, 0, and 0, 1. And the idea is that there is different clusters in these neurons. Some of them are cortical spinal tract. Some of them are more reticulospinal tract. There are different inhibitory, excitatory kind of like functions or for each of these clusters. if you want to really read more, so you can go to that paper and then look at the how we defined and organized this network. But the idea was that we really followed the very classical study in monkeys stuff that where they did kind of like controlled lesions to these or different tracts, going down from the cortex to the spinal cord. One of them was a study back in '68 where they actually lesioned the cortical spinal tract at the midbrain level, and they observed, this is a video, original video from the study by Lawrence and Coopers in '68A, showed that monkeys can really jump. They still have a lot of force in their hands, but they cannot really perform any of these dexterous finger pinch precision movement to grab that apple food pellets and to eat it. So this is one of the studies that showed that, well, this actually could be, this tracks very important for fine motor control, but nothing to do with the more powerful and strength, forces that the monkeys need to apply, because there's no deficit on that aspect of the behavior for the monkeys after this controlled lesion on the cortical spinal tract. Right. So the same group, they went to a different tract, and they cut the reticulospinal tract again at the medial level, and they observed that the monkeys, become very weak. They lost a lot of their power and their forces, but still the monkey was able to perform this kind of like very fine motor control of finger dexterity. All right. So these were the first two studies showing that it might be there is a different roles of cortical spinal tract and reticulospinal tract in different aspects of the hand function. One is more biased toward fine motor control, dexterous movement, and one biased toward more strength and force production of the movement. All right, so there's some dissociable things about these two tracks, and this is exactly what Ashraf did in this network. And just to summarize that, we were able to reproduce the behavior measures after performing lesion in different areas and in different sizes. So this is what's interesting in computational, that you can apply stroke wherever you want. So you basically delete that neurons from the network. So you train the network to perform this normal behavior, and then you make a lesion to the specific different areas, and then you can make a lesion with the different sizes of that area, and then you can test the outcome of that specific network to the behavior. And so one of the nice prediction of that artificial network, again, this artificial network, one of the nice prediction is that this is the individuation ability pre-stroke. So the network really can replicate the behavior that we observe in the human. So we have a really good individuation ability in the normal level. Then we apply the stroke here, and then you can see really a reduction of that individuations. And this reduction was a function of the severity of the stroke happening in the network. All right, up to 40% of the cortical spinal tract lesion affected a little bit of the individuations, but these networks actually still reproduce a very nicely behavior in the output. All right, mid-size, a little bit more than mid-size, a lesion between 50 to 70% suggests that, well, there is a really initial large reduction in our individuation ability after this kind of stroke. But the network can still recover after that stroke, but they never actually goes back to the same baseline level here. So there is a big gap between baseline here and recovery after 50% stroke versus 70% stroke of the cortical spinal tract. Right? Much larger lesion to the cortical spinal area, 80% and above. The network predicts that it will never recover after this kind of stroke. All right. So this is something that we need to verify it in the clinical population and the clinical stroke patients. And this is also very interesting here from the rehab perspective, because if we really understand that larger lesion load to the cortical spinal tract would never allow the system to recover and go back to any level of dexterity, so then we need to think about alternative compensation ways to achieve that dexterous movement, and rather than trying to train the impaired hand to make that movement. right? This is something that it's not like, it's more about personalized rehabilitation per participants based on some quantification where the lesion and how big is the lesion affected the cortical spinal tract. Right, this is something that is actually nice prediction from this model that we can test it. Okay, so then we can go now after the lesion, and we can go to all of these kind of like activity and weighted weights between these kind of like different levels, and we can investigate how the weights change after the stroke and how they change after the recovery. Okay, and just to make it a really conclusion here is that we observed that in the artificial neural networks, so many of these survival or residual neurons are actually participates in the recovery. Even the reticulospinal tract that I showed before is very important for force, not for dexterity and fine control. These kind of like networks show that it actually they change their activity, the weight change after the stroke, and they somehow participate of the the recovery of the finger dexterity, right? Now, is that true for humans? We don't know. But we know there is a few studies, actually, one study published 2 years ago or 1 year ago, when they recorded from monkeys in this reticulospinal formation, doing some cortical lesions, they actually observed there is a lot of change that happened in other areas that we did not think they were really important for dexterity movement, while the monkeys performing finger movement. And it seems there are some plasticity changes happening on that, I would say, different areas that somehow involved in the recovery of the monkey after lesion to the cortical spinal tract. All right, this is something also that the network can predict. All right, so the last part of my talk about more about it's a new study that I'm really excited about is to look at biological neurons, real neurons that were really active during finger dexterity. And this is a collaboration between our lab and Mark Schieber from Rochester University and Daniel, my students. We look at activity of a few hundreds of neurons in the primary motor cortex of 2 monkeys. This is the location of these neurons and the depth and the position coordination of these neurons. And the monkeys were instructed to perform a finger individuation task based on a visual cues that they see in the screen, and then they trained to produce a finger individuation task. This is the displacement of the thumb movement, the index, the middle, the ring, and the pinky when the monkey instructed to perform that specific movement. So they train very well, so they can perform very nicely this kind of like finger individuation task. So we made sure that the monkeys can learn the task, and then we started to look at the neural activity. And then we started to look at the neurons. It's just a big mess. It's really hard to make any conclusions about activity of specific neurons that were tuned for a specific aspect of the behavior. So there, you cannot really find any neurons that can fire only for a specific digit. It's really hard to find any neurons that can fire for a specific direction or a combination of these. So I will just, for showing this kind of thing, this is 2 different neurons here. Well, this is one neuron that is really highly active. This is the raster plot, like the spikes, and this is the histogram of these spikes. So it's really active, and this dashed line is the movement onset. So it's really active in movement of the index in the extension. So 2E is index extension, but he's also very active in the 3 extension and the 5 extension. He's less active in the 5 flexion, but he's active in the last part of the movement, in that flexion, flexion of the fifth finger. If you go to the different neurons, you will see that this neuron in some tribes actually it's actually more active pre-movement and gets silent after the monkey starts the movement, and there's an EMG recorded from that. So there's a lot of diversity, variability across single cell activity that can explain if there's any tuning toward any specific function of that finger. But then the nice thing is that it comes when we actually recorded EMG activity of that neuron. So we know we can we can know if what is the target with the target muscle of a specific neurons. And we do that by a classical spike trigger averages effect. All right, so you can like average across all spikes, and then you can define neurons if they are sending facilitation to their target muscles or they're sending inhibition to their target muscles. This is average of EMG activity of their target muscles, and you can quantify that things, and then you can cluster your neurons based on their post-spike facilitation or post-spike suppression, and trying to understand what is actually the role of that kind of things. So what we start to observe is that there is a very interesting effect of post-spike suppression. Neurons in the cortex, in the premotor cortex, that send suppression to their target muscle in the antagonist muscles when they move muscles that need to be somehow suppressed. So they tend to do that before the movement onset. So this is one of the functions of that CM cells during finger individuation movements. All right, and this was significantly, we can observe it in the population, and all of these neurons together, and also we can measure EMG. We see a little bit of inhibition happening of these in the agonist, in the antagonist, and the stabilizer muscles. There is a quite small inhibition happening before that movement onset. All right. But then again, as I said, like this is really one aspect of the function and the role of these CM cells. But if you look at the population level, you see, as I said, a lot of things happening. And there's a new theory, actually, I would say, suggested to explain this kind of like variances and variability in the neural level, even during the preparation activity. So there is no really any movement that you can measure. There is no really any EMG muscle activity, but the neurons are really active in that period. So what they are doing, what kind of like computation are doing. So this kind of like is the dynamical population activity, a framework, where you can explain, well, all of this activity during preparation is actually about in a space that is not really your output space or potent space or EMG space. It's a null space that it is not random. It's actually doing a lot of computation that preparing your next movement when the trigger comes and when you actually start to produce EMG activity in your output space. All right, this is from Krishna Shinoy, people from Columbia University and Stanford University, where they actually started to explain this kind of like theory in monkeys doing reaching movement. So we started to do this kind of like population level analysis, and with that I will finish. Is that there is a lot of information even at the very low dimensional neural trajectory spaces. We can really identify and detect digits. Characteristic, like which digits are going to move, and different colors here, which direction between flexion and extension. And if you really look very closely, you will see that most of this activity you can already quantify and clusters before the movement onset. So if you see there's some blue dots here, this is the black dots. All of this activity before the black dots is actually before the movement started at all. So you can easily differentiate between direction and digits when we start to look at population activity. And a lot of this information actually comes from neurons that are active before the movement onset. So in general, and like in total, all of this activity driving our specific digit movement. And I will end with that. So I really focus a lot on the primary motor cortex, M1, CM cells, and some of the premotor doing some lesion analysis. But I believe this is not the full story, right? I think this is, this is the classical view that I just was showing you today, that there's a lot of activity happening in the motor cortex and the cortical spinal tract that drive motor neurons to kind of like activate different muscles. And some of our artificial neural network kind of like hint to some change in the reticulospinal formation together driving this kind of like movement. But I think there are also a lot of subcortical areas involved with this finger dexterity. One of them is the cerebellum, and there's also other things. And this is because there is a very recent study showing that I think they are from Cleveland Hospital. They showed that DBS to the cerebellar dentate nucleus actually can improve hand function and finger functions in stroke with subcortical and subcortical lesions. So the cerebellum is somehow involved. Yet we don't really fully understand what is the mechanisms that driving these changes and how we can, program some rehabilitation to directly target these subcortical and cerebellar areas in terms of like finger dexterity. This is another non-human primate, but I just will skip it. It's also about showing some evidence that cerebellum is really involved in some aspects of finger movement. So this is a collaboration between with Yvette Prout. She developed a very nice high frequency stimulation where she can block the cerebellar output. So the flow of the commands sending out to the motor cortex from the cerebellum while the monkey is performing finger movement and dexterous task. And then we compare the kinematics and the deficits, impairment in controlled condition versus high frequency stimulation, where we reversibly block the cerebellar output to test the role and causal role of cerebellum in finger dexterity. And that's it. I would like to first thank all of my students that are doing all of this great job, the collaborators and the funding agency for supporting us. And thank you so much.